we have today Dr. Hanno Steen. This is also a pleasure to have you here, Hanno. And I would like to introduce you. So you actually are now at the Boston Children's Hospital, and you're also affiliated with the Harvard Medical School. In fact, you're associate professor of pathology at Harvard Medical School. And, it's really interesting to see you're the director of proteomics and, you're working in the neurobiology program, but also the precision vaccine program. And it's really interesting to see these combinations. And more recently, you have been quite interested also in immunology projects. I could go on and on with all the things that you're involved with, but then I thought, well, maybe we'll let you talk to us about this. And the title of your talk is indeed high throughput plasma proteomics, something of high interest to a lot of us. So thank you, Hanno, for making the time to be with us today.
Okay, thank you very much for the kind introduction, Birgit. And yeah, I guess originally, actually, Steen was invited to present here as part of Best of HUPO 2023. Fortunately, Jules couldn't be here, so she then asked me to present on her behalf. And I have to say, the previous story was really cool, this levitation. I always think it's cool. So in contrast to this, to that story, this is really kind of a down-to-earth problem that we had here in the lab that I'm presenting right now. Let me just see whether, there we go. So, I'm in the, as Birgit mentioned, I'm in the hospital. And when you work in a hospital environment, you can't really avoid working on body fluids. And you kind of also get all kinds of cohorts when you talk to people, when people talk to you, and you start somewhere with one sample with kind of small sample numbers, 3 and 3 or whatever, but eventually you kind of really notice that 3 and 3 really just doesn't cut it any longer. You really want to move up to the kind of tens of samples, and that's exactly also what happened in my lab over time. We kind of evolved from single digits to 2 digits. Then kind of things got more exciting. We moved to hundreds of samples. And that's also then the moment when we decided, listen, we can't really work on single tubes any longer. Everything should be done in 96 well plates. And in that context, we published 8 years ago this, what we call the Meston blotting platform, which in our hands worked very well. It was a pretty cost efficient, pretty fast way of processing 96 samples in one swoop. So that really worked very well. But I think now with the new technologies out there and also with the demand out there, we always have to keep in mind that proteomics in many ways has been the kind of slowpoke of the omics, that often enough there's this, oh, we would like to do this larger study, but we don't have enough money. So we kind of do then 10 and 10 samples. So I think there was really the interest and the need to kind of move to larger numbers. And so larger numbers here, 1,000 samples, that was in a project we got involved in where we had 5 years time to analyze 1,500 samples, which was kind of at that time was a pretty decent number. Then word of advice, if you go into this kind of large scale sampling, once you get here to these large sample numbers, you really want to pay attention to your sample management also. So you really individual tubes and kind of handwritten labels, single channel pipettes, everything kind of gets a little bit a nuisance when you deal with these large sample numbers. So it's kind of really this number is when you want to start thinking about barcoding, barcode readers, multi-channel pipettes, even liquid handling robots and so on, just as a word of advice. And so that's basically where we tinkered around in the late 2010s. And then basically, COVID fell out of the sky in many ways. And so at the beginning of COVID, when many labs were shutting down and temporarily at least, and we were also already preparing to shut down, the NID approached me and to become part of this impact study, immunophenotyping assessment in the COVID-19 cohort. And it was a nationwide study with 14 clinical centers, 7 national core sites. And anybody who has interacted with an IED, that, they ask you for help, you say yes, and then you worry about the details later. So that's exactly what we did also. And the cohort or the impact study itself was actually very interesting. They want to enroll hospitalized COVID patients. And please keep in mind that this was beginning of the pandemic. You couldn't really get any healthy controls because healthy people wouldn't go to the hospital, or you weren't even allowed as a healthy person in the hospital. So there's always something you have to keep in mind here. And so they want to take 6 samples during the hospitalization, and then 4 samples 12 months after, up to 12 months after convalescent. So all the details can be found here in the Science Immunology paper. And from our perspective, again, it was a immunophenotyping study. So they really want to do a lot of different things with these samples, and different samples, serum, whole blood, endotracheal aspirates, nasal swabs, and you can see here all the different omics and all the different assays they want to do. And all these assays were supposed to be done here at the various different core sites, and my lab here was in charge of proteomics and metabolomics. For metabolomics, we were more the clearinghouse, we were organizing it, but everything was then shipped out, whereas the proteomics was supposed to be done in my lab. But here comes the kicker, and that was something, you know, you're asked, you say yes, but then you kind of only, when you think about it, you kind of realize, oh boy, I'm not sure whether that was a good idea. Because our aim was to enroll 1,000 patients and to have up to 10 samples per patient. So when you do the math, that you realize, whoa, they're asking me to analyze 10,000 plasma samples. And you can go into the literature, you will not find any paper on plasma proteomics and 10,000 plasma samples. So, okay, well, after the initial shock, we kind of rolled up the sleeves and got moving. So basically here, when you go from 1,000 to 10,000, this is basically how it looks. So it's a lot of samples. So we still have 2 -80 freezers full of samples. And it's, yeah, it's quite a daunting task to dig through them and sort through them and so on. Anyway, because it was beginning of COVID, it's not that we could hire new people and so on. It was more we pivoted. Everybody in the lab kind of dropped the projects they were involved in, and basically got involved in this large-scale COVID project. And basically everything we did then, we got done with these 6 people here. Kinga and Meenakshi were doing the sample and project management. Then Benoit and Arthur, 2 French postdocs in my lab, were basically in charge of the plasma proteomics. David Stevens was the only person who joined my lab during COVID. We called him Mr. SP3. I don't think there aren't too many people out there who can say that in 2 years in a lab they have processed 15,000 plasma samples. That is David's claim to fame. And then Patrick, he kind of had joined my lab to do wet lab proteomics, and then became our computer geek. Anyway, so with these people, with this excellent team, we moved forward. And I should mention that we had 3 months time to develop a pipeline and lock it in, and so that we were ready to receive the samples. So the impact study, we ended up here, and we got samples in 5 phases, 4,500 samples, 63 plates, and that amounts to, in case you're wondering, 4 months of 24/7 instrument time. When it comes to plasma proteomics, you always have to deal with, or you always have to think about this aspect, or this kind of image of the iceberg. You only kind of iceberg, you see a little bit above the water, but there's so much more underneath the surface of the water. And this is the same with plasma proteomics. When you basically do neat plasma proteomics, you only see always the same 300 to 400 proteins. And on the other hand, we have to also, because it was an immunophenotyping study, we had to analyze these very abundant proteins, because they're very important to define the immunophenotype of a person during health and disease. On the other hand, we also did want to look at the kind of what's underneath the surface of the water, basically, or kind of the tissue leakage proteins in plasma. Just to contextualize the problem here, again, 20% of the most abundant proteins account for close to 90, or account for almost all the protein content. When you do just a neat plasma proteomics, you identify the same 300 to 500 classical plasma proteins. You have really hard time to get down to the tissue leakage proteins. So people have devised those depletion methods, and most of them are antibody-based. But antibodies by default were slow, expensive, and they do lack robustness in that regard. That anybody who has worked with antibodies knows that just small changes in pH, small changes in soil concentration is going to affect affinity and specificity. So we knew that this is kind of a non-starter when it comes to analyzing thousands and thousands of plasma samples. So, and Arthur, luckily Arthur was already kind of working on a old method from 1948 that was published and actually every so often popped up in proteomics for enrichment of histones, for the enrichment of tau in blood, for enrichment for peptidomics. So every so often kind of this method popped up, but evidently it wasn't really that widely used. But when Arthur was playing with it, we actually did notice that this method is really, really great to get rid of the most abundant plasma proteins. So that was basically then the pipeline. And when you do, again, high throughput proteomics, always keep in mind, you want to have the method as simple as possible. So we got the plasma samples, kind of 96 well plate compatible. And then with this perchloric acid precipitation that Arthur made compatible for high throughput proteomics or plasma proteomics, you basically add the perchloric acid to a final concentration of 3.5%. You let it stand in the freezer for half an hour, you spin it down, you take the succinate, and then you go on from there. And so it was really a method that kind of lent itself for high throughput because it was really simple, very easily implemented, and could be done by anybody. And yeah, the question is then really kind of, does it work? Yes, it works. Here's a correlation for the plasma protein rank before depletion is on the x-axis, on the y-axis after depletion. And you can see here that almost all the top 20 proteins are going away from this diagonal, meaning they are depleted. And even here, for instance, albumin goes from rank 1 to rank 45. So you can do the math. It probably translates into depletion of 99.99% of albumin, but because it's so highly abundant at the beginning, you still easily detect it. But it's not the most abundant protein any longer. It's, as I said, beyond rank 40 at that moment, giving you an idea. Then transferrin also, rank 125, fibrinogen gamma also. Then also here is, for instance, one protein, normally highly abundant, rank 24, 23 in plasma, but when you then deplete it, it goes to rank 400. So it really is very efficiently depleted. Then it is cheap. So we're talking about reagent cost 2 to 3 cents, depending on the volume of the perchloric acid you buy when you do this. Then you have to do a little bit more cleanup because you deal here with a super acid. It is highly acidic. So you have one extra step of cleaning up. We use the HUPO plates from waters, but I'm sure that other plates work as well. It is very reproducible, and that's kind of something we're always proud of this plot. There's a PCA of 3,300 samples analyzed over 10 months in 3 batches. And I hope you agree with me that there's really no batch effect, no discernible batch effect. So that really kind of speaks for the robustness of this method. And then also we had on every plate, we had some, what we call universal reference standards, and which allowed us, we had 6 universal reference standards on each plate. And that kind of gives you an idea also that the CVs were easily in the 10% range. We had only kind of 2 plates where we had some issues, but otherwise it really worked very well. And then it really allows also for deep plasma proteomics. Kind of this was exemplified, for instance, that we could readily detect SARS-CoV-2 derived nucleoprotein with sequence coverage above 20%. So this isn't a kind of a single peptide barely detected. This was solidly identified. And please keep in mind that SARS-CoV-2 isn't a blood-borne pathogen. So there are a couple of papers out which I described that kind of either target methods on detection of endotracheal aspirates and so on, but not necessarily in plasma. So we were quite proud that we could even detect these proteins or this particular viral protein in discovery plasma proteomics. And in the latest iteration, we identify now more than 2,000 plasma proteins at 60 to 100 samples per day. That's our throughput that we are operating at. And one way to look at it also how deep can be deep is taking just all the reported FDA-approved biomarkers. And when you don't deplete, then you get this kind of box and whisker here for the concentration of the known FDA biomarkers, and you're here in the range of 1.4 to 42,000 nanograms per milliliter. But then when you deplete, you kind of open up and allow for the detection of much lower concentration. Now you're down to 0.2 to 120 nanograms per milliliter of kind of protein concentration, just to give you an idea that you can really dig very deep with this method and see much, much more than you would see otherwise. So the method itself was published now 6 weeks ago in Science Advances. It's open, that paper can be downloaded by everybody. So if you're interested, go into the webpage and download it. We're quite excited that in 6 weeks, it was already downloaded more than 6,000 times. So it seems we hit a nerve here that people are interested in high throughput plasma proteomics. So this was then basically what we ended up doing here. For the classical plasma proteomics, we just developed a high throughput, highly multiplex MRI method, because again, you know exactly what the most abundant plasma proteins are. You don't have to kind of rediscover them again and again. That went on to triple quad. And then for what we call the extended proteomics at that moment was a depletion that went then onto the EvoSep with the timsTOF Pro 2. And that was really, from our perspective, a winning situation, winning combination. It's great hardware, really robust. Then when you do these large scale studies, then the question is always, how do you analyze large data sets? That was a problem we didn't really expect it. We thought we were well off with the hardware that we had in the lab, but actually turned out that even the hardware we had in the lab didn't allow for the efficient processing of these large data sets. Just to also contextualize this problem, before, and we ran Q-exactive instrumentation, various generations. And you're happy when you run 20 samples a day. Each of them is 500, or kind of half a gigabyte to gigabyte each. So you talk about 10 to 20 gigabytes per day. So when you talk about the timsTOF and high throughput mode, you're talking about 60 to 100 samples per day. You're talking about around 2 gigabytes each file. So you're talking about more than 100 gigabytes per day. And, as I told you, that we needed for the entire study 40 days of instrument time. So you can do the math and figure out how much data we have generated. We are talking here about terabytes. And again, here we dealt with unprecedented sample numbers, unprecedented data amounts from a proteomics perspective. And so we reached out to a couple of companies, couple of groups, and they all kind of shrugged their shoulders, told us they couldn't help us, but everybody also then added, well, if we find a solution, we should talk to them because they would be interested in hearing more about it. And so luckily at that time, at the beginning of COVID, the Massachusetts Green High Performance Computing Center went online. We were one of the first users here, and Patrick, even though he doesn't have any formal comp-sci education or informatic education, he became our computer whiz. He was working with a research computing in Boston Children's Hospital. It's one of the first users of the MJH-PCC cluster. And he developed then a pipeline, kind of highly parallelized pipeline to analyze this large data set. This is then kind of interesting also to see what the different kind of improvements are doing. One kind of major part was the parallelization of a lot of the processes. I should mention that not everything could be parallelized, but a lot of it could. But then also because this kind of this analysis of the IO heavy operations, just moving everything on a solid state drive next to the CPU basically gave you another factor 10 in speed. So I'm not talking about kind of network access somewhere in the cloud. I'm just talking really kind of moving it not to a hard drive next to it, but really to a SSD, a solid state drive next to the CPU, GPU, in order to allow for efficient processing. Was interesting for us also to see that that makes such a big difference. So also, if you're interested in that, the study was, this particular set of work was published last year in JPR. So feel free to check it out. So with that, I would like to conclude my talk. So basically, from my perspective, high throughput proteomics is now finally here and is also here to stay. Then the whole platform that we have developed, we have by now also kind of tested and established for various body fluids. We have done in urine, CSF, tracheal aspirates, saliva, oral secretion, you name it. And, I've been in the field for kind of far too long, you can argue, and proteomics has promised for many, many years or almost decades by now that we will discover a lot of biomarkers. But, you know, our success rate isn't that great, admittedly. But I think now with high throughput proteomics, we can finally now use those very large scale or large cohorts in order to really do meaningful plasma biomarker discovery. And yeah, from our perspective, we provide here this deep plasma proteomics pipeline, which has been by now applied to 15 to 20,000 samples. So what we published was kind of the method fully validated on 3,500 samples. And right now, I should mention also that we have done everything on a single instrument. It's not that I could increase the throughput just by having 10 instruments standing there. We did everything on a single instrument. So we have this kind of rhythm now where we process 2 plates per day, every other day, and then analyze those samples at 60 to 100 SPD samples per day on the instrument. And when you do these large-scale work studies, robustness is a must. It's really key. Without a robust setup, it's really hard to approach that. So in our hands, the EvoSAP1 and the timsTOF Pro is a winning combination. So it really worked extremely well in our hands. And the timsTOF Pro, also because of the EvoSAP, only needed deep cleaning after 5,000 plasma samples. So we were really impressed by that, I have to say, especially since plasma is really a really dirty body fluid sample type. And also when you do the data analysis, it does require high performance computing environment. So please keep that in mind. Anyway, with that, I would like to acknowledge the people here. The lab, it's a joint lab with my wife, Steen, who was supposed to speak, but as I said, she's airborne right now. And one of my key collaborators, Ofer Levi, the people in the lab. And I would like to highlight again the people who kind of pulled it off at the beginning of COVID. And just kind of 6 people plus 2 instruments really allowed us to establish this pipeline and do it all. And then also the funding agencies and the consortia I'm involved in was the largest funding for this particular project coming out of the NID. And with that, I would like to thank you, and I'm happy to answer questions.
Yeah, well, thank you, Hanno. This was excellent. I mean, amazing tour de force that you described for us there. I love the combined lab that you have. It seems very powerful in many directions. Yeah, that was actually one of my questions with the cleaning. You answered it, right? That's actually quite remarkable after 5,000 samples, because that's always a concern, right? Does it, I mean, everything goes super fast, but then there's a lot of downtime. That doesn't seem to be a problem. That's really great. And also your variance seems really good. I mean, so what do you think, so say if now you have automated a lot, imagine if like a person does it more on a more smaller scale where there's more individual person involvement versus doing it with a robot. Do you think that could still be robust enough then, or?
Yes, it does. So we still have a couple of people who have smaller cohorts and they kind of, I'm not going to use a handling robot. I do it myself manually. And, you know, the nice thing is with this perchloric acid, it's because it's such an absolute condition you use, you just add kind of, I don't know, you use 70% perchloric acid, you end up with a final concentration of 3.5%. So it's really the initial salt concentration, initial small variation in pH doesn't really matter. It's really kind of, it just works beautifully in our hands. And, you know, as long as you put in the freezer, it's always the same temperature in the freezer. You do it for half an hour and then you just pipe it off the supernatant. It's you know, the beauty of this workflow is also it's so simple. And I think that was really also one of the key aspects that we learned when we did this, that simplicity is key. And, you know, I talked about robustness is key, but simplicity equals robustness in many ways. So when we started out, we initially thought also about, for instance, doing TMT labeling just to increase the throughput, but then we figured out, you know, it's just too complex. And then you also, at that moment, you will have to start thinking about to control the yield each time you do a TMT labeling and so on. And that just kind of, it just makes it far too complex. At this moment, we decided, you know, brute force, simple workflow is just at that moment gives us so much more than trying to go fancy.
No, that's great. I love that. Yeah, no, it's really true, right? Simplicity, you know, to robustness. You had a 9% variance that you showed in one of your slides. That's really good.
I have a question.
Yeah, please, Jeff.
Hanno, that's really cool. That's amazing. I'm curious, like, how deep do you think you guys are? Like, do you think in some, at least a handful of these samples, like you've identified nearly all the proteins that are detectable or even present?
I think there's, well, first of all, talk to the people from SomaLogic. They say, oh, we can identify 7,500 proteins and plasma.
Yeah, that's all false positives.
Well, I'm not going to say anything. I'm a lawyer, but so, you know, to be honest, I think it's a little bit like, so how many proteins do you find in a cell? And you can always argue, you know, there are always some cells that kind of go bust at any given moment. And so you can argue that you should be able to identify every single protein, you know, at the ultimate constant sensitivity and so on. But the question is, and at the end of the day, you know, when you do this high throughput, so people have asked me, so how many proteins do you identify when you do 1 hour gradients or when you do this or that? But, you know, our objective was we were supposed to analyze 10,000 samples on a single instrument. So at that moment, high throughput was key and robustness was key. So we haven't really actually at this moment, and only now we are starting kind of going back and saying, hey, how much can we do when we do half-an-hour gradients, when we do 1 hour gradients? But we haven't done, or we are in the midst of that work, but that was an objective of this. And, you know, high throughput is always, it's a compromise. You can't get it all. You know, there's no such thing as free lunch. And again, if you have 10 instruments standing there, obviously you have many things you can do, but with only a single instrument, at that moment, you do the math and you realize you have to analyze at 60 or 100 samples a day in order to get anywhere. And at this moment, you know, we're quite happy with those 2,000 proteins at that throughput. And we are actually more excited about now approaching large cohorts than trying to get out of smaller cohorts more proteins. But that's kind of in many ways a philosophical question. And then it also depends a little bit on the disease you're worrying about. If you have a very rare disease where you have only 10 and 10 samples, you really try to get the most out of those samples, and then obviously it makes sense to do our gradients. But, you know, we have another study where just a discovery is 400 samples, a validation is 500 samples. So it's kind of very empowering to be able to take on those 1,000 samples and just get them done and not worry about kind of burning a student there or postdoc for his or her entire PhD or postdoc time. Just as an example, I have now visiting a PhD student from Germany, Mohammed Osman, and his project for the 6 months is, one of the 2 projects is to analyze 1,500 plasma samples. You know, in the old days, that was impossible feat, but that's now what we are doing in the lab. Again, it comes at the expense of probably analytical depth, but that is not the question right now we have.
Yeah.
Awesome. Makes sense. I completely agree.
I agree. Well, it also allows, as you pointed out in your study also, but it allows for longitudinal dynamic assessments, right? And I feel really the longitudinal strategy with biofluids has like real value, right? Because you have the same person where you get multiple time points, and so it's wonderful to think about that in the context of your study. Yeah, wonderful.
I actually have a question. Hanno, that's a wonderful talk. This is Tom Gao from UIC. I have actually 2 questions. One related to what Jeff just asked. So you have like all these like samples. That's like, you know, amazing, you know, resource of all the proteomics data. Have you guys actually tried to actually improve that identification by something like a match between run? Because I think that'll be very interesting to see how match between run actually works on like, you know, thousands of samples. You know, before people have been applying this for like, you know, 10,
no,
5,
it's actually, I should mention that. So there's a little bit of a story to it. So first of all, with the match between runs, we did everything with match between runs, and that was really also what initially killed our efforts to use our in-house hardware. And I should mention that even though we are now doing it on a high performance computing cluster, even kind of, you know, analyzing those 4,500 LCMS runs with match between runs takes 3 weeks compute time because of the match between runs. Without match between runs, things would be much, much easier actually. And so kind of just to address that. And then the other question that also, you know, it's often DDA, DAA. So while we started out with DDA, by now everything in the lab is DAA based. And, you know, there obviously it depends on how good the spectral library is. I personally still a fan of spectral libraries. Obviously, kind of in special situations, we also just use a spectral library free approach. But if you have a spectral library, we use a spectral library, and it definitely helps to solidify your searches and your identifications.
And just one more question. How you actually share these files like over the internet? For example, like, you know, how long does it take to actually, you know, deposit this into Prime?
Yeah, no, it's actually interesting, you know, when you deal with those things, everything takes so much longer, and it's really kind of high throughput or kind of scaling up. It's a different animal, you know. It's also when you optimize the method and you just save 1 minute per run at that moment, you know, when you have 1400 runs, it already saves you 1 day of instrument time because a day has 1400 minutes or 1440 minutes. So, and also, you know, if people ask, so how long does it take? You know, just take your cell phone and try to download 10 gigabytes of photos. And now kind of think about that 1,000 times, because that's kind of what we talked about 10 terabytes of data. And this is one of those things, you know, in the old days, or I transfer data kind of, you know, you start before your lunch break, you go for lunch, come back, and then it's done. Whereas here, you know, you leave on Friday evening for the weekend, you come back on Monday and you hope that half the data transfer is done. And then you also have to worry about, you know, that you have to double check that really kind of nothing is corrupt and so on. So everything is kind of, you have to be a little bit more careful. So again, here talking here about days and days for data transfer, not kind of minutes or hours. So everything, and that also means that you actually plan a little bit more ahead instead of just starting and, oh, this went wrong. Oh yeah, right, stupid idea, do it again. No, here you really kind of, you sit down for half an hour and then you go ahead. And also, for instance, I mentioned kind of briefly alluded to the sample management. That was something we learned the hard way, that when you deal with these large sample numbers, sample management becomes incredibly important, because when you screw up there, everything is bad. And so you have to be very careful there. And we were, as I mentioned, the clearinghouse for metabolomics for proteomics. So any sample we got in, we had to actually aliquot 7 times. So it basically took us 2 weeks just to come up with the aliquoting scheme, because we only want to freeze or thaw the samples once and then do all the aliquoting and sub-aliquoting into all the different tubes and plates and so on, because each platform had its own preferences for tubes, for plates and so on. And then kind of what's then also interesting, you know, the most annoying part for the people in the lab dealing with these samples was to open and closing the tubes. You know, it's kind of funny, those little things that are normally kind of, oh, you don't worry about it, but when you do it in these large sample numbers, it actually becomes a nuisance. And even though there are those really cool machines that kind of screw and unscrew 96 tubes in one go, it's still it takes a long time. And then, yeah, you can buy those kind of elastic mats that you kind of pop in. But, you know, especially when you do medium or long-term freezing, they are not as good as proper screw caps. So that's why we opted for the screw caps instead of those kind of little silicone mats. So, you know, when you deal with those large sample numbers, a lot of things are very different.
Yeah.
Thank you so much.
Thank you very much.
This was really amazing and very insightful for us, and a wonderful way to close our webinar today that we had with, I mean, I think very inspiring to hear again. And so, yeah, thank you, Hanno, so much for jumping in very, you know, like on short notice. And Jeff, I wanted to thank you also, not only for the webinar today, but you were actually one of the co-organizers of the main conference, right, Jeff? And I hope you enjoyed this day too.
It was great. Thank you for allowing me to participate.
Wonderful. And thank you to Conference Solution and to Lydia, and thank you to all of our audience. And again, this will be posted on the webinar and on the website. And yeah, so please come all to US HUPO 2024. All right. See you all there. Bye-bye.
Thanks, everyone. Thanks, Brigitte.
Bye. Thank you.